Hello everyone, welcome to this video. This video will produce the lab procedure for particles 2, which is particle 2A, 2B, and the particle 3. Particle 2A is spectrophotometry, reducing sugar analysis using the 3,5-dinitrosalicylic acid DNS methods. Particle 2B is determination of sugar in sample using the 3,5-dinitrosalicylic acid DNS method, and the particle 3 is the physical properties of different lipid and fatty acids. Okay, this is our group members. The first one is Alia Aviana Sarawak anak Lubak. The second one is Alisa Joanna anak Nyau. And the third one is Anissa Chendu anak Jinam. And the last one is Felicity Lemah anak Banyang.
Next, we will move to the procedure 2A. Firstly, we carefully dispense a glucose solution for each of the test tube using 1000 micropipettes. For tube 1 is zero point 0, for tube 2 is zero point 0.2 milliliter, for tube 3 we dispense zero point 0.4 milliliter, for tube 4 we dispense zero point 0.6 milliliter glucose solution. We dispense zero point 0.8 milliliter glucose solution into test tube 5. Lastly, we dispense 1.0 milliliter glucose solution into test tube 6. Second step, we dispense distilled water into each of the test tube using 1000 milliliter micropipettes. For tube 1, we dispense 1.0 milliliter distilled water. Next, for tube 2, we dispense 0.8 milliliter. For tube 3 is 0.6 milliliter. For tube 4, we dispense 0.4 milliliter. And for tube 5 is 0.2 milliliter. Meanwhile, for the test tube 6 is 0.0 milliliter. The next step was adding 1 milliliter of DNS reagent to each of the test tubes. After we transfer all of the solution in the label tubes, then all of the test tube were mixed by using a vortex mixture. After all mixed well, we place all of the tubes into a hot breaker and boil water for 5 to 7 minutes. The last step is we cool all of the tubes. After a few minutes, we add 1 milliliter of Rochelle salt, then transfer the solution into the cuvette. Then mix well using a vortex mixture. Then all of the samples were analyzed using a spectrophometer. Now we will move to the procedure 2B. First, we put a sample water in 5 test tubes. For the first tube, put 1000 milliliter of sample water. In the second tube, we put 500 milliliter. In the third tube, we put 250 milliliter. In the fourth tube, 100 milliliter. In the fifth tube, we put 50 milliliter. And in the last tube, we did not put any sample water. After that, we put distilled water in the sixth tube. In the first tube, we leave 0 milliliter. And in the second tube, we put 500 milliliter of distilled water. While in the third tube, we put 750 milliliter of distilled water. In the fourth tube, we put 900 milliliter. Then in the fifth tube, we put 950 milliliter. And in the sixth tube, 1000 milliliter. Next, we put the DNS region in sixth tube, 1000 milliliter. Next, we put a glucose solution for each of the test tube using 1000 micropipettes. After we transfer all of the solution in the label tubes, then all of the test tubes were mixed by using a vortex mixer. After all mixed well, we place all of the tubes into hot breaker and boil water for 5 to 7 minutes. Then we cool all of the tubes in 5 minutes. After a few minutes, we add 1 milliliter of Rochelle salt, then transfer into the cuvette, then mix well using a vertex mixer. Then all of the sample were analyzed using a spectrophotometer.
Let's move to the practical tree. In practical tree, we are given with five materials to do this practical. First one is microcentrifuge. Second one is dropper. Third one is distilled water. The fourth one is toluene. And the last one is the three selected lipids. In this practical, selected lipid that has given to us is the best of food, which is the vegetable oils, shortening, and butters. Firstly, we prepare two separate sets of microcentrifuge tubes, each containing 3 tubes for a total of 6. Label each tube carefully using permanent marker to ensure clear identifications throughout the experiments. The first sets will be labeled A1, B1, and C2, and the second set as A2, B2, and C2. Each letter A, B, C represents a different lipid containing product that you have brought with you to the lab. The next steps involves testing the solubility of the lipid containing product in a polar solvent distilled water. This part of the experiment we perform using sets 1 tube A1, B1, and C1. Using clean dropper, carefully adds 1.0 milliliter of dropper to each tube in sets 1. Next, to test the nonpolar solvent toluenes, we added 0.5 milliliter of toluenes to each tube in sets 2, which is A2, B2, and C2. Then we cap the tube and shake gently to mix. Once capped, gently shake each tube for 5 to 10 seconds, then mix the contents thoroughly using a vortex mixer. Then observe what happens after shaking. Lastly, after make the content truly using a vortex mixer, we make an observations on lipid rations about the appearance color, odors, and the solubilities in water and toluenes. After we complete all the solubility tests, we record and observe the results in the data tables. Throughout all of the verticals, we have learning a few new things. From this lab, we know how to use micropipettes wells. And if it didn't use in the proposed way, it may impact the data and accuracy of the data will be compromised. In this practical, we also challenge face incorrect data that we need to repeat it again, and we have to face the time management too because the time that's provided for us is very limited. My suggestion improvement is I hope there will be advanced technology in pipette so that data will accurate and didn't need to face the challenge to incorrect data that we need to repeat it again. That's all from me and my teams. Thank you for your attention and hope to see you again.